Hi, welcome back to BIO4YOU. Okay, so we are moving on to chapter 6.2. In this video, we're going to discuss gene mutation for 6.2. So first, we're going to look and define the gene mutation, define the 4 types of gene mutation, and describe the consequences of base substitution such as sickle cell anemia. So mutation, there are gene mutation or point mutation. There are 4 that we need to study. The first one is base substitution, base insertion, base deletion, and base inversion. Okay, we will look onto details on each of these mutation after this. So for chromosomal mutation, we will study on this on 6.3. Okay, so gene mutation is a change in base sequence of DNA molecules that represent a gene. So when it involves only a single base, it is called a point mutation. So maknanya kalau ada berlaku satu saja mutation, satu saja nucleotide base, a single base, kita panggil sebagai point mutation. This will lead to the change in amino acid sequence. Okay, remember when we read amino acids DNA sequence codon by codon. So each codon will encode for a specific amino acid. So when the codon is being wrongly encoded, i.e. having mutation, mana berlaku mutation, so bila codon tu salah, amino acid tu pun akan salah juga. And thus may change the proteins or enzymes that is intended to be produced. Sepatutnya hasilkan protein lain tetapi protein lain yang terhasil. Why? Because susunan amino acidnya sudah berubah. The different protein produced as the effect of mutation may not function as normal due to the mutation. Okay, so base substitution, namanya pun substitution. So it's a replacement of one nucleotide and its partner with another pair of nucleotide. Maknanya kita tukar saja. Bukan kita tukar lah. Maknanya berlaku pertukaran nucleotide tersebut. Base insertion, maknanya addition, tambah nucleotide pairs in a gene. Base deletion, loses, hilang nucleotide pairs in a gene. Base inversion, reversal of 2 or more nucleotides in a gene. Okay, first we look at base substitution. So this is the normal DNA with the normal sequence AGC-TTA. So after substitution, T is being substituted with G. Okay, so we'll see how this will affect. Alright, so this occurs when one nucleotide is replaced by another nucleotide, and this will result change in codon during transcription and will result change in particular amino acid during translation. Okay, we look here. This is the normal sequence for a normal gene, and this is the mutant DNA. So at this sequence, so sepatutnya after AAA is the nucleotide with C. So substitute T instead of C. Maknanya sepatutnya C tapi dah tukar menjadi T. So sepatutnya CCG will encode for glycine tapi TCG will encode for serine. So sepatutnya glycine amino acidnya sekarang dah jadi serine. So itu adalah base substitution. Next is base insertion. Maknanya kita masukkan one nucleotide base dekat situ. So kita masukkan after AGC sepatutnya TTA, but here after AGC ada C pula nucleotide yang masuk. Lepas tu baru TTA. So of course lah C partner dia akan dengan G. Okay, it occurs when one or more nucleotides are added into sequence of DNA nucleotide. This will cause frame shift mutation. So apa frame shift mutation ni? There will be a change of all amino acids at and after the point of insertion. Okay, remember that we read codon by triplets of the sequence, triplets of the nucleotides, AUG, CGA, UUA, UAC, GGG, and etc. So if we add after UUA, we add one more U. So what happened is that the next codon will be UUA. So instead of it encodes for tyrosine, now it encodes for leucine, UUA. And then you can see the next codon will be read by as CGG. Dia kat sini CGG. Sebab apa? Sebab dah tambah satu U dekat depan ni. And G yang akhir sekali dah termasuk kepada next codon. So berlakunya shift, frame shift mutation ni. So masuk frame shift mutation ni, frame codon selepas-lepas ni dah berlaku shift. So that's why it is called frame shift mutation. Okay, so when we heard this frame shift mutation, or when this frame shift mutation occurs, kita dah tahu efek dia adalah sangat besar ataupun sangat teruk. Kenapa sangat teruk? Sebab starting daripada point of mutation tu sampai belakang-belakang tu semua jadi haywire, semua jadi rosak codon tu semua jadi salah. Hanya sebab tambah satu sahaja. Okay, so this diagram shows you what happened for the base insertion. So base G is inserted, and you can see the encoded amino acid sepatutnya serine, glycine and lysine. But then now jadi serine, nampak starting after point of mutation kat sini, belakang-belakang ni semua jadi proline, jadi glutamic acid, semua dah berubah dah. Okay. Okay, so this one we see this is before insertion and this is after insertion. So we have here CCU, AAA, CCU again here. So after the insertion of G nucleotide after U, so what happened is that after CCU now become GAA and ACC because ACC, and this U has been jumped onto the next frame, next codon frame. So there's frame shift mutation that occurs here. Okay, so caused by base insertion. So next one is base deletion. So similar like base insertion, when you add one nucleotide, but for base deletion we remove one nucleotide, one pair of nucleotide. When we remove T, also eventually we will remove A as well. Okay, so this is the normal DNA, AGC-TTA. Then you remove this T. So what happened is that AGC-TA, then it will continue with the next codon. Okay, it occurs when one or more nucleotides are removed from sequence of DNA nucleotide. This will cause, again, frame shift mutation. There will be change of all amino acids at and after the point of deletion. So the example that you need to study is thalassemia. Okay, penyakit thalassemia. Deletion of nucleotide base pairs for genes which control the production of hemoglobin alpha or beta chains. Okay, so ini adalah thalassemia. Okay, so this is the normal DNA strand. Okay, after transcription, you have the encoded mRNA here, and after the process of translation, we would expect it will encode amino acids serine, glycine and lysine. So however, after base T is missing, T ni hilang. Okay, so bila T ni hilang, after transcription dia akan hasilkan mRNA yang ini. And through the process of translation, after serine dia jadi arginine, serine dan seterusnya lah. All amino acids starting from this point will change, akan berubah. So in this slide, we look at the base deletion. So this is before deletion. GAG, GUU, CCU, AAA, CCU again. Okay, so after the deletion, when this A is being deleted, so AA, this C will goes into the will join this codon, so it become AAC. Okay, so instead of lysine being encoded, so now it's asparagine is being encoded. Alright. And frame shift mutation. Frame shift mutation will occur if the number of nucleotides inserted or deleted and is not in multiple of 3. So kita baca codon tu secara triplets bertiga-tiga. So kalau kita hanya tambah satu, shift yang belakang tu frame yang belakang, frame of codon belakang tu semua akan terganggu. Sama juga kalau kita delete. Tapi let's say if tambah 3, which is rare, baru belakang tu tak berlaku frame shift mutation. Sebab codon tu still dibaca dengan bertiga-tiga. Okay, resulting the triplets codons are wrongly read from the point of mutation and differ from that of the normal gene. This has a very serious effects because it causes all amino acids beyond or after the point of mutation to be changed, and this producing different proteins. So this diagram will show you what is the effect of frame shift mutation. Okay. And next is base inversion. Okay, inversion ni bertukar tempat lah. Okay, so this is the normal DNA. The sequence is AGCTTA. So what happened is that C and T, they exchange place, invert. So nampak C and T bertukar tempat. Okay, so this will not cause frame shift mutation because dia tak ada, the codon still be read in the same triplet. Dia tak kacau codon-codon yang belakang. And when base inversion, when two or more nucleotides exchange place and the sequence is reversed. Inversion of nucleotide do not have a very serious effect. Dia hanya ada efek pada paling kurang dua amino acid yang terlibat sahajalah. Okay, so this diagram shows you the base inversion. So this is the normal DNA strand and this is the mRNA. So these are the encoded amino acid. Okay, however, after the base inversion, so these are the amino acids being encoded. So only this amino acid is changed. Maknanya yang tengah-tengah ni sahaja yang berubah. Yang lain semua tu masih kekal sama. Serine sama, lysine pun masih sama. Sebab tu dia kurang serius sedikit. Okay, so this diagram, okay, this was cuma nak bagi gambaran kepada kita, kepada korang, macam untuk fahamkan konsep pasal base substitution, insertion, deletion ni. Katakanlah kita ada ayat, the fat cat ate the rat. Okay, so ni bila berlakunya base substitution, T tukar menjadi R, so kita masih lagi boleh baca, tapi hanya kodon ini saja yang mengalami pertukaran lah. So pada CAT jadi CAR sekarang ni. Okey, ni hanyalah analogi sahaja. Kita of course lah tak ada kodon H, tak ada kodon E, tak ada nucleotide T, H, E, tak ada. Ni hanya gambaran untuk ayat sahaja. So kalau base insertion, okay, the fat cat ate the rat, ni yang normal. So bila kita tambah satu dekat sini, the fat cat, kita tambah C dekat sini, dah yang sini jadi cat lagi pula. Lepas tu belakang-belakang ni kodon ni terus menjadi berubah. So frame dekat frame of codon belakang-belakang ni akan berubah. This base insertion will leads to frame shift mutation. Same goes with base deletion. Kita delete F, so dia dah jadi ATC. Ni ATC to be the next codon. So belakang-belakang ni semua dah jadi codon-codon, dah jadi codon yang berbeza dah. So dah tengok, kita tak boleh nak baca dah pun ayat ni. So that's how serious this base deletion also leads to frame shift mutation. So for base inversion, tukar tempat sahaja, the fat cat ate the rat. So bila tukar tempat sahaja, dia punya kesannya taklah teruk sangat because it only involve the particular amino acid sahaja. So now we're going to look at the 3 effects of gene mutation. So apakah efek-efeknya of gene mutation ni? So dia ada 3. Yang pertama adalah missense mutation, second one is nonsense mutation, and the third one is silence mutation. Okay. So the first one, missense mutation. Okay, missense. Why is it called missense? Okay, miss ni from the first word kat sini, for example, kita try to aim at something, we're shooting, we miss, we just only miss 1 point, we just miss one target. So this results in the change of the one amino acid with another amino acid during protein synthesis. As a result, the activity of an enzyme or hormone might decrease or be destroyed. Ni kesan kepada protein dihasilkan tu lah. So example dia adalah sickle cell anemia. Okay, so we look at this diagram. So instead of we are expecting to have amino acid methionine, lysine, phenylalanine, glycine, but we have the first 3 correct. Only 1 is wrong because due to this change of nucleotide. So instead of glycine, we get serine. So missense, kita hanya dapat satu saja yang tidak tepat, we missed it. Okay. So the missense mutation, this one is normal. So CTT will encode for glutamic acid, whereas CAT will encode for valine sebab T dah tukar menjadi A. Okay. So this is the example of sickle cell anemia. Instead of glutamic acid yang dihasilkan, valine yang dihasilkan. So this is normal hemoglobin. This is S hemoglobin, simplified for sickle cell anemia. So sickle cell anemia, thymine is substituted with adenine in the gene which encodes for beta chain of hemoglobin. So codon for glutamic acid becomes codon for valine. Sepatutnya kita dapat glutamic acid, dapat valine. So abnormal hemoglobin will crystallizes and erythrocyte is pulled into a sickle shape. So sepatutnya bentuk tu biconcave, dah jadi bentuk sickle cell shape. So this is how the shape of the sickle cell red blood cell looks like, and this one is the normal red blood cell. Hanya tukar satu amino acid sahaja, glutamate tukar menjadi valine. So dah menyebabkan this serious disease. So patient who suffers from anemia will have these symptoms. So the abnormal hemoglobin, bentuk dia macam ni, sickle shape. Hemoglobin is inefficient of transporting oxygen, okay, because the shape, okay, is not normal, so that tak boleh efficient transport oxygen. Organ damage due to insufficient supply of oxygen, ialah dah tak cukup oxygen kan, and also physical weakness. Okay, next we have nonsense mutation. Okay, nama dia pun nonsense. Kenapa nonsense? Okay, because it will encode for a stop codon. Changes a codon for an amino acid into a stop codon. So translation will be terminated prematurely and produce short polypeptide and lead to non-functional protein. Okay, nonsense mutation. Sepatutnya saya expect ada 4 lah amino acid yang sepatutnya encoded here. Okay, you always need to remember this, the stop codons, which are UA, UAA, UAG, UAG, and UGA. So when we encounter this stop codon, the translation process will stop and no amino acid will be encoded afterwards. So sepatutnya kita expect 4, now ada 1 je. So that's why dipanggil sebagai nonsense mutation. This is nonsense lah. I'm asking for 4. Why I only get 1? So nonsense mutation. Again, why? Because U is being A here is being substituted with U, so you have UAG. So this is a stop codon. So again, ACC now become ACT, and the mRNA is UGA. Can remember there are 3: UA, UAG, UGA. So UGA is a stop codon. And last but not least is the silence mutation. Apa maksud silence mutation ni? Although the codon is changed, it has no effect on amino acid sequence because it code for the same amino acid. Macam ni pula tu. Okay, we look at this diagram. GGC encode for glycine. Okay, U is being U instead of C, maknanya ada berlaku pertukaran lah. U menggantikan C. However, GGU also encode for the same amino acid glycine. Okay, how can this happen? Okay, remember if you refer to the table of codons, okay, one amino acid can be encoded by several types of codon. So maknanya untuk amino acid glycine ni boleh encoded by GGC, also GGU pun juga. So dekat dalam hal ini kita panggil sebagai silence mutation. Sebab apa? Amino acid yang dihasilkan tu tetap sama. So sebab tu silence mutation. Silence tu maksud dia tak ada efek pun, tak ada kesan pun. Walaupun ada berlaku mutation, macam tak ada apa-apa perubahan je. Sebab amino acid yang dihasilkan tu masih lagi sama. Okay, that will be all for this second video for chapter 6. Please stay tuned and watch for 6.3, and please do like, subscribe and share with your friends. Okay, until we meet again.